Welcome everyone, and thanks for joining our workshop about how to design and optimize a full spectrum high dimensional immunophenotyping panel. And also thanks for being here on a Sunday. I'm Laura Ferrer, and I'll be one of the presenters today alongside Sam Small. Sam, she's a senior staff scientist at the Hugh Green Cytometry Center at the Malagan Institute in New Zealand, and her role focuses around supporting researchers in all things flow cytometry related, from training a range of users from beginner to advanced, to panel design, data analysis, and troubleshooting, cell sorting, and other ongoing support. She's passionate about ensuring high standards are maintained when producing flow cytometry data. And me, Laura, I am the high dimensional cytometry specialist at the Hugh Green Cytometry Center at the Malagan, also in Wellington, New Zealand. And I am highly interested in full spectrum high dimensional cytometry and its applications to immunophenotyping and in new biological contexts. I am a member of ISAC Data Standards Task Force and an ISAC SRL Emerging Leader since last year. Sam and me work together at the Malagan, and we both are science and cytometry education lovers. At the Malagan also, we were first adopters of the Aurora, and this was 4 years ago. And soon we became fascinated about the potential of this technology. So today we have 4 Cytec Auroras at the Institute. The workshop content for today, it's going to be first, we will introduce you to full spectrum high dimensional panel. Next section will be about panel design. Next one, panel optimization. And the last section, we will talk a little bit about longitudinal experiments. We will have 2 breaks, one of 10 minutes and one of 5 minutes. The first one after the panel design section and the second one after panel optimization section. And please, we've seen that there's no Q&A section. So if you can ask your questions in the Zoom chat, we will answer them at some time that we have allocated to answer questions. This workshop aims to be interactive, and for this reason, we will use a Slido to run polls and quizzes related to a case study. So if possible now, we would recommend you to use a secondary device for a Slido, such as your phone or a tablet, or if you have a double screen computer system, this will work too. Otherwise, if you only use one screen, it can get quite slow and confusing to keep changing from Zoom to Slido and vice versa. So now we'll give you some time to connect to Slido, and please let us know through the chat, Zoom chat, if you have issues, and we will try to help you. So a couple of minutes, maybe. So is it working for everybody, Slido? Yeah. Anyone is having issues? Shall we continue? Cool, so we will continue. In case you have issues, let us know. Perfect. So since 2017 that we got our first full spectrum flow cytometer, we have published several papers about full spectrum flow cytometry. And for today's workshop, we thought that we could benefit of the content of these 2 protocol papers that we publish in current protocols. The first one was published in 2020, and it's about how to design an immunophenotyping panel, while the second one has been published this year, and it guides you through about how to optimize the immunophenotyping panel. And also now it's time to introduce you our friend Charlie, and we will stay with Charlie during all the workshop, and he's very happy to meet you. Charlie is a PhD student in our institute and he's studying T cells. He has been asked to design and optimize a high-dimensional flow cytometry panel to run on the Cytec Aurora available in his flow core. We think he may need your help during this workshop, so please be ready to help him. And at the end, we can have a small celebration if he can finish his PhD. We'll see. So yes, if there are no questions about how everything is going to work, let's just start by briefly introducing full spectrum flow cytometry to you. Here we have the example of P, and this graphic is illustrating the emission profile of P in a conventional cytometer. On a conventional cytometer, that probably some of you are more used than a spectral flow, the fluorescence emitted from P is utilized in only one, so from only one bandpass filter. In this example, I'm talking about bandpass filter 2, while any light in neighboring bandpass filters will need to be compensated out, and all the other portions of the spectrum will be disgregated. In comparison, when we assess P on a Cytec aurora, we capture light from the entire emission spectrum. In this way, we capture so much more information from every single fluorophore on every single particle. Interestingly, we capture all that information from every laser that is present on the aurora we are using, excluding only the portions of the spectrum which coincide with the laser excitation frequencies. So for every laser on the aurora, there is a matching module of light detectors. In this way, we not only collect emission from the entire spectrum from the primary excitation laser of a fluorophore, we collect the emission spectrum of that fluorophore from every laser present in the aurora. In this slide, the top plot most closely resembles the P emission profile shown on the previous slide that was about conventional flow, with the box channels outlining the portion of the spectrum characterized by the conventional cytometer. Have a look that when we characterize P emission from every laser on the system, in this example, we have 1 laser compared to 3 lasers and 5 lasers, we gain much more information about how P is emitting fluorescence, and correspondingly, how this fluor is likely to contribute a spread into other fluors in a multicolor assay. This profile is the unique signature of PE, so we are no longer limited to using just one fluorophore in one bandpass detector. We can use highly overlapping dyes, and still we will be able to resolve well populations of interest. For example, take P and C-fluor BYG584. These fluors have highly overlapping spectra, and assessed on a conventional cytometer would be incompatible in a multicolor panel. But when we look at their unique spectral signatures, we find that there are a number of areas in which they are different. And this difference enables us to spectrally unmix them and use them in the same multicolor panel, and with very, very careful panel design, even use them in markers that are co-expressed. Spectral unmixing, another important concept. Spectral unmixing involves informing the cytometer about fluors present in the assay with the use of single stain reference controls. And then the ordinary least squares algorithm is used to calculate the contribution of each known fluorophore spectra to the total collected emission signal. So have a look at this 3-color mix sample, and we extract or deconvolute each component until nothing is left. If you have noticed four lines in this animation instead of 3, it's because you have visualized also the autofluorescence spectral pattern, and we will talk more later about the autofluorescence pattern too. Another important introductory concept is what does high dimensional mean and what does involve. FCS files with lots of parameters will be high dimensional. What is a lot will be though up to you. It can be 10 parameters or it can be 40 parameters. The classical strategy for flow cytometry analysis is gating on 2D plots. But the higher the number of parameters in your panel, the more iterations are possible. See in this slide an example of a 20-color panel with which you may end up with 200 2D plots. This is not only time consuming, it is also not efficient in terms of getting conclusions because there is too much information in your data. So high dimensional data analysis will aim to simplify and summarize your data and will help you obtain meaningful biological results. But the important thing is that the quality of your panel is going to be vital. Here, I would like to show you one example to verify that the Aurora data has enough quality to run high dimensional data algorithms. And we compare its performance with Cytometry, the gold standard technology at that time. So this was 4 years ago. In case you don't know, Cytometry uses, instead of fluorophores, metal tags conjugated to different antibodies targeting your markers. There is almost no spectral overlap, what makes panel design easier. But the acquisition usually is much more slower. 2 panels that had shared markers were run. And when using UMAP, that is an algorithm that reduces high dimensional data to 2 dimensions, we were quite surprised to see that our UMAPs were looking very similar and that we could identify the same populations in both data sets. We could also identify some differences. So for example, XCR1 positive events are defined better in the data collected by a spectral flow cytometry. But overall, the analysis looked very, very comparable. The results were also verified using FlowSum clustering algorithms. And we also found that both technologies' analysis looked very similar. The all-important message to take home from this slide is that the data is going to look very comparable, but we need to invest time in panel design and panel optimization to make sure that the data has enough quality. And now I'll pass over to Sam, who will guide us through how to design a full spectrum high dimensional panel.
Okie dokie. Hi, everybody. So the first thing you need to know regarding spectral flow cytometry and any flow cytometry at all is that magic doesn't exist. If your panel hasn't been well designed and optimized, don't expect high quality data out of it. I'm showing here one real example of one of our users that came to us to help them with their data analysis. And just taking a look at their compensation matrix, it's quite shocking because it's absolutely terrible. I hope you already know what we kind of expect to see in a compensation matrix, but in case you don't, here is a better example. So we're expecting to see orthogonal stainings and that the negatives are clean and tight. We don't want to see any banana shapes. So focusing on panel design is absolutely essential. Here we have one reference about high dimensional panel design. And last year, we also published a protocol of the steps to follow when designing a spectral flow cytometry panel. For this talk, I'll guide you through all of the steps in this figure on the right. So step 1, determine the experimental question. Following that, we need to know the biology. We need to know the instrument and the spectral signatures. We have to assign fluorophores to each marker. We then have to review the theoretical panel design, and if it is well designed, we can optimize it at the cytometer. So here comes Charlie, our PhD student, in our first case study question. So Charlie's supervisor has asked him to create a large panel with as many T-cell markers as possible, but doesn't give him any targeted analysis goals. Charlie is happy with this and sets out to make a list of all of his favourite markers, being sure to include a few to rule out the unwanted cell types. The list starts looking quite long, and Charlie thinks such a big panel might be quite hard to design. But he was asked to add lots of markers, so he keeps going. In the end, he has come up with a list of 34 markers that he thinks should be in the panel. Is he missing something important? So if you head over to Slido, we'll activate our first interactive poll. So we're going to create a word cloud. If you come up with some ideas of an important experimental aspect that Charlie should be using to guide his panel design. And I'll give you 2 minutes to to put in some answers.
One more minute for more suggestions. Chad is very happy. He's getting lots of suggestions. Thanks for helping him.
Okay, so let's take a look at the answers that we've got. So there's a bit of variety, but one theme that is coming up quite a bit is a goal, a biological question, a hypothesis, and their research question, which is exactly what we were hoping you guys would guess. Yes, so step number one for any panel design is to know the experimental question. There's no point in throwing every marker at the wall and trying to design a panel that is way too big if you can narrow it down and figure out exactly what you need to answer with the particular markers. So when determining an experimental question or multiple questions, we need to consider the scope of the question. Are you addressing a very specific question, such as are distinct CD8 T-cell phenotypes found during different treatments, or a broader question, such as does the distribution of cell populations differ between conditions? The scope of the question may dictate the parameters that are measured. Are we interested in cellular abundance and expression profiles, or identification and quantification of cellular diversity, or maybe changes in population structure? Also, some metrics to consider how to answer your question should be determined. Which readouts are needed to test your hypothesis? Which readouts are more critical than others? For example, are you looking at MFI or counts or something else? Which tissues will have to be analysed, and what are the limitations associated with that tissue? For example, digestion or tissue stability or autofluorescence. And how will you need to process the samples to obtain the readout of interest? Will you need to fix them, permeabilize, or stimulate to get the answer you need? So Charlie needs to come up with a question to shape his panel design, such as in patients treated with drug X, is there a shift in ratio between different types of cytokine-producing T helper cells? Or after prolonged stimulation, which T cell subsets are most prone to exhaustion? That way he can narrow down his panel a little bit. The most critical part of panel design is knowing the biology. The only way to have successful panel design is to have sufficient information on the biology of the assay. If you don't know this information, you can either ask someone in your lab who has more knowledge regarding the project or look at the literature. But we really recommend you to invest enough time in this step to gather as much information as possible. So Charlie is back. He's finalized his list of markers once he's got his experimental question, but he realizes he doesn't know too much about T cells. He's fresh in his PhD. So he turns to his lab mates for help. and he asks them all about the exact cell types each marker is on and how abundant that marker usually is on those cells. And he gets a good amount of information about most of the markers on his list, but he has a few markers that are unique to his project, so the others don't have any experience with them. Charlie's excited to start his panel design, so he decides to carry on with the information that he has. Besides, it's only a few markers without information. That won't screw up the whole panel, right? So over on Slido, we have our second poll, and we want to know where else could Charlie find marker information. So what are your favorite places to get information about markers for your panels, or where would you go? We'll give you 2 minutes to fill in some suggestions for Charlie. We've got some great ideas coming through. Keep them coming. One more minute. All right, we've got some good ideas for Charlie here, and a lot of them follow a very similar theme in that we're mostly going to go to the literature and previously published panels, a couple of votes for OMIPs, which are a great source, as well as vendor websites can sometimes have information about marker expression. So yes, that's some great ideas for Charlie. He knows that he doesn't have to just rely on his lab mates for this information. It is imperative to know which markers are necessary to accurately define the cell types of interest and which markers are important for the functional readout necessary to address your experimental question. We first need to note the cell populations that we are interested in and need to be analysed. For example, CD4 or CD8 T cells. Secondly, we need to annotate the lineage markers necessary to identify the cells of interest. For example, with CD8 T cells, we'll use CD45, a leukocyte marker, CD3, a T cell marker, and CD8 to specify CD8 T cells. And lastly, we need to define the markers that are to be used as a readout to address the experimental question. And now it's a good time to think about which antigens are absolutely essential. You need these to identify your specific cell populations, and which antigens are non-essential. These are bonus antigens that might provide interesting information, but not critical to your hypothesis. And the information that you mainly need regarding the biology is the level of antigen expression and if the antigens are co-expressed on the same cells. We need to understand the classification of each antigen. This classification system was described by Yolanda Mank and Mario Roederer in 2007 and involves assigning antigens to 3 main classes: primary, secondary, and tertiary. Primary antigens are those with high density and exhibit on or off expression, for example, CD3, CD19, or CD14. Secondary antigens are those with intermediate density and often exhibit continuous expression patterns, for example, CCR7, HLA-DR, and CD38. Tertiary antigens are those with low density or unknown expression. This is often the category to which you will assign the antigens in your panel, which will be associated with your experimental question. It is also vital that we understand the co-expression patterns of each antigen in the assay. The antigens which are mutually exclusive and those which are co-expressed will have varying impacts on our panel design. Use the literature to establish your understanding of the co-expression patterns of as many antigens in your panel as you can. For example, in this gating tree, CD45RA is only expressed in naive cells, while CCR7 is co-expressed in naive cells and central memory cells, so they will have different impacts on the panel design. To facilitate the collection of this information, we suggest generating a table which includes columns to record the markers you are interested in, note the expression levels of each antigen and their co-expression, decide on the clone to be used, clones based on previous experience or publications, and search for the availability of fluorophore conjugated antibodies for each marker of interest. And fluorofinder.com is a great website to obtain all of the fluorophores available from each company for a single marker. Once we know all of the biology, we then need to know the instrument we're going to be working with and the spectral signatures of our different fluorophores. The panel will need to be designed while taking into account the configuration of the machine you'll be using. The configuration is the number of lasers and detectors that one machine has. Both Northern Lights and Auroras can have different configurations depending on the instrument, ranging from 1 to 5 lasers. Also, a 4-laser Aurora could have 2 different configurations. In the case of Auroras, we don't need to worry about the detectors, only the laser configurations. Charlie's back. He has his list of markers and his marker information. So he is on the hunt for a spillover spreading matrix, so he can use spread information in the panel design process. Charlie isn't sure on the configuration of the Aurora in the facility, but he does find an Aurora spread matrix for a 3-laser system. And he starts assigning fluorophores to the different markers. So over on Slido, we have a multiple choice question. And I'll give you 1 minute to pop in your answer for what might be the result of using a spillover spreading matrix that doesn't match your actual cytometer configuration. All right, so it looks like 75% of people got the right answer, which is that some fluorophores will be fine, but others will be affected by having different lasers. So as you can see here, that the different spillover spreading matrices are actually different. So for most of the matrix, things are actually relatively similar, but in certain regions, you will find areas where increasing the number of lasers would increase the spread of certain fluorophores. And if everybody wants to have a go in the chat of answering why they think adding an extra laser might increase the spread of certain fluorophores, that would be cool. Does anybody have any ideas? We've got one idea of cross-laser excitation, which is pretty close. Added complexity. So the main reason that adding a laser will increase the spread is that you're suddenly, oh, hang on. Well, the main reason we see increase in spread when you add a laser is you're suddenly exciting your fluorophores with their actual primary laser. So here in this example, we're going from a 3-laser system, which doesn't have a yellow-green laser, to a 4-laser system, which does. So suddenly all of your PE-based fluorophores are going to be excited a heck of a lot more optimally once you add that yellow-green laser in. And as you increase your brightness, you also increase spread. So that's the main reason as to why we expect more spread with an added laser. But it doesn't affect those fluorophores so much which aren't primarily excited by the added laser. Moving forward to spectral signatures, knowing the spectral signature of each fluorochrome should also be considered. So fluorophores that have the same spectral signature can't be used in combination in the same panel. Only dyes with unique spectral signatures that will yield distinct patterns after unmixing should be used in the same panel. Dyes with similar spectral signatures can be used at the risk of introducing spreading error, which can be mitigated if they are used with markers that are not co-expressed. The spectral signatures of many fluorophores have been extensively defined, and this information can be found in the Cytec or BioLegend spectral viewers, making sure that you're selecting the spectral viewer for BioLegend, not their regular one. And we never design panels without consulting spectral viewers. When designing your panel, attempt to disperse your fluorophore peaks as widely as possible to reduce overlap as much as possible, which will help to reduce fluorochrome spread, something we will return to a little bit later. Aside from the online spectral viewer, the spectral signatures of a huge number of fluorophores for each configuration can also be found on the Cytec website in their fluorochrome selection guidelines. One tool to use to know how similar two fluorophores are is the Similarity Index. The Similarity Index is the measure of uniqueness between two fluors. An index of 1 means that the spectra match exactly, and an index of 0 means that the spectra are completely different. For panel design, the more unique a set of dyes are, the easier they are to use together as there is less spillover. In the first example on this slide, we see PerCP Sci 5.5 and PerCP eFluor 710, two dyes that are highly overlapping. The similarity index between these two dyes is 0.9. In the second example, we observe BUV 395 and APC. Note that in any channel where BUV 395 is emitting light, APC is not, and vice versa. So these two dyes have a similarity index of 0. At this point, we are ready to start assigning fluorophores to the different markers in our panel. And here I'll introduce Charlie and the panel that he is designing. And he has got one slot left right at the bottom of the table that he still has to fit a fluorophore into. So we'll go through all the theory of assigning fluorophores, and at the end, we'll see if we can help Charlie out. At the time of selecting fluorophores, there are some important pieces of information that we need to know. We need to use unique fluorophore spectral signatures. We need to consider the brightness of the fluorophores that we are planning to use. And we also need to be aware of the spread that our fluorophores can cause to the other fluorophores in the panel. Before we start talking about each of these considerations, I would like to briefly say that knowing the autofluorescence of your samples before designing your panel can reduce potential issues in your panel by designing around this autofluorescence. In highly autofluorescent tissues, it is recommended that before designing your spectral panel, you first run your unstained sample to know the autofluorescence signature, or if there are multiple autofluorescence signatures. Then we can think about the autofluorescence as kind of like another fluorochrome, and therefore think of distributing our markers with considerations for the autofluorescence. We can think about putting bright markers in the high autofluorescence space, which will draw out of the spread of the negative, allowing you to still spread out your fluors and use the low autofluorescence regions for tertiary markers that need protecting. To make sure that we select unique signatures, we have the online resources I mentioned, as well as the Spectrum Viewer and Similarity Index tool on the Cytec website. The stain index is a very useful measure to be able to determine the resolution of a fluorophore. It is the ratio of the separation between the positive and the negative population divided by 2 times the standard deviation of the negative population. If another fluorophore produces a lower median fluorescence intensity, or MFI, for the positive population than the previous example with the same MFI background, its stain index is going to decrease as the separation between positive and negative is reduced. Also, if another fluorophore can produce an MFI for the positive population similar to the first example, but has a larger variation in the negative population, even with the same MFI, its stain index is going to be reduced. This makes sense as it is not as good at resolving the 2 populations as the first one. The next characteristic we need to understand about fluorophores is their inherent brightness. Cytec assessed brightness by calculating the stain index of each fluorochrome when conjugated to human CD4. So in this way, the changes in brightness are attributable to the fluorophore and not antigen density. Each of the fluorophore guides includes a stain index of fluorophores to help you with this process. In this chart, the fluors are listed in order from dimmest on the left to brightest on the right. And when deciding which fluor to assign to each antigen, it is best to assign the antigens with highest expression to dimmer fluors and those with low expression to brighter fluors. The third consideration is the property of a fluorophore that will have the biggest impact on your panel design, and that is how much spread it contributes to other fluorophores in the assay. In this example, fluor Y does not spread into fluor X, but fluor X spreads into fluor Y, and this spread reduces the stain index of marker Y and thus reduces the resolution, the ability to separate positive and negative. The spread is used to calculate how much the stain index of the single stain sample is reduced by the spread introduced by another dye. Quality panel design is a balancing act aimed at managing fluorophore spread. As well as the Spectrum Viewer and Brightness Index tools already mentioned, you can also find a cross-stain index in the Cytec fluorophore guides on their website. To read this table, the spread of the fluorophore in the row impacts the resolution of the fluorophore in the column. Red means the fluor in that row has significant spread into the dye in the column. Areas in bright pink and red indicate pairs for which more attention to panel design is needed. So when we look at the spread of APC into BUV, 805, 0% spread is observed and all populations are well resolved. Conversely, when we look at the spread of FitC into Spark Blue 550, 76% spread is observed. And as a result, the negative population is significantly spread. In order for us to manage this spread, these 2 fluorophores have been assigned to mutually exclusive antigens, whose expression patterns are well resolved from the negative. So now we are ready to start assigning fluorophores. The most difficult task in multicolor panel design is to match the most appropriate fluorophore with each marker of interest. As I've previously said, spread, fluorophore brightness and availability, and the levels of marker expression as well as marker co-expression have to be considered. In order to facilitate the selection process, we suggest the following steps. Define fluorescent reporter genes first, as they are the least flexible. Next, assign very rare markers to the only fluorophore options available and common markers to rare fluorophores. Match the fluorophore brightness with antigen expression levels by combining brighter fluorophores with weaker expression levels and vice versa. Assign the remaining markers to other fluorophores considering their co-expression and spread. Co-expressed markers and markers selected for functional readouts should be matched with fluorophores that receive minimal spread. And due to the range of available options, the viability dye can be selected last, as positive cells will be dead ones and we don't want to interrogate them further. At this point, we can use the spectral viewer to make sure that our fluorophore choices are well spread across the whole emission range and have unique spectral signatures. Also, we can have a look at the similarity indices to see how similar our fluors are, and also at the complexity index. The complexity index is an overall measure of the uniqueness of all dyes in a full spectrum flow cytometry panel. A lower complexity index will translate to an easier combination of dyes to work with, while a higher number means that the combination of dyes is getting trickier to work with. The complexity index is a useful tool for assessing fluorophore selection when designing the panel. Values will vary depending on the fluorophores used in the panel. A high number for a 10 colour panel won't be a high number for a 30 colour panel. In our case, 13 was a reasonable number for a 23 colour panel on a 3 laser aurora, but as said, 13 would be a very high complexity index for a 4 colour panel, for example. So here is an example of a 10 colour panel and how we can impact the complexity index by bunching up our dyes into specific regions of the spectrum. With spreading it out, we can lower our complexity index down to 2.4 or increase it all the way up to 16 if we push everything into similar regions. And if we increase the number of colours in our panel, you can see a nice low complexity index is 23.2 with everything well spread. But if we bunch all of our fluorophores into only a couple of regions of the spectrum, we can make our complexity index shoot through the roof to 146.2. So the goal here is with the number of colours and the configuration you have, you want to decrease your complexity index as much as you can. And here are some examples to visualize similarity indices and the complexity index in a 2-color panel. So the higher the similarity of 2 dyes, the more difficult it is to separate them, which causes distortion of the negative. As you can see, the aim is to have the lowest complexity index you can manage for your panel. The last step recommended before jumping onto the instrument is to QC the theoretical panel design. This consists of making sure that our designed panel is what we think is closest to the one that will work very well in reality. So do a double check. Review the panel on a marker by marker, cell by cell basis, looking at whether the fluorophores that induce high amounts of spread are on non-co-expressed markers. The readout markers receive a minimal amount of spread, and fluorophore brightness and antigen expression levels are well matched. If the answer to these 3 questions is yes, then we're getting a lot closer to optimizing our panel on the instrument. And here we have a panel coded to show primary, secondary, and tertiary. And note that tertiary are on very bright fluors, primary are on dimmer, and secondary in between. These 3 markers down the bottom are on the same laser line and are neighbours. So they have to be put on different cell types. So we've got Siglet F on eosinophils, CD64 on macrophages, and monocytes, and CD90.2 on T cells. If potential issues are found here, it's a good idea to try and fix them now by making panel design rearrangements rather than pushing forward to the cytometer. Okay, so now we know the basics. We're going to use the spillover spreading matrix and stain index rankings on the next slide in combination with the panel details that Charlie's already put together to figure out which of the following 4 fluorophores would be the best fit for FOXP3, actually it's CD28, in Charlie's panel. So we've got 4 options of fluorochromes here. And then you're also going to answer the question, what made the other 3 choices less suitable? And then once we've discussed the answers to those 2 questions, we're then going to take another look at Charlie's panel and decide if any of his fluorophore assignments that he's already made are too bright or too dim. So here we have the panel that Charlie has started designing, and we've got 1 slot left at the bottom for CD28. And then we have 4 fluorophore options for you to choose from, and using the spillover spreading matrix, remembering that fluorophores in the row cause spread to fluorophores in the column, and the stain index ranking to decide which of these 4 fluorochromes would fit best into this slot in Charlie's panel. So we'll activate the poll in Slido, and we'll give you 10 minutes to work through Charlie's panel and come up with answers. So the answer for which fluorophore should fit into the slot, and why would you not pick the other 3 options. So jump on Slido and fill those in, and we'll get back to you soon. Any last minute takers for question 1? Give you a few more seconds before we move on to question 2. Alrighty, so we got 60% of answers saying APC, 7% saying PE, no one voted for BV510, and 33% saying AF647. All right, before we tell you the answer to this one, we'll activate question 2. So we want to know, why did you pick your fluorophore and not the other 3? We'll give you a few minutes to answer this one, and then we'll discuss our answers. Alrighty, let's discuss the answers. So we're getting quite a few ideas, but a lot of them are along the right lines. So we've got a lot coming up around spread, fluorophore brightness, antigen expression levels. So people are using the right kind of ideas for picking their markers. So if we go back to the multi-choice question, most people answered APC, which was indeed the marker, the fluorophore that I would put into this slot. And my reasoning behind this, which a lot of you picked up on, so why wouldn't we pick PE? It's a really nice bright fluorophore. It's not introducing any spread. It's not receiving any spread. But it's already in our panel, so we can't reuse PE. APC, the one I picked, it's a nice, decently bright fluorophore. It's only contributing spread into Percy-P Psi 5.5, but that doesn't matter because we're putting it on a low expressed fluorophore, and the one that it's spreading into is a very highly expressed marker on CD8 T cells, so we don't need to worry too much about that. BB510, can't pick that one. It's way too dim to be on a low expressed marker. And AF647, it's a little bit brighter than APC, but it receives a heck of a lot more spread than APC does. So putting it on a low express marker means that we're going to run into a bit more trouble if we've got lots of markers spreading into it. So we might lose a lot of our resolution. So that is my logic behind picking APC for this marker in Charlie's panel. So we have a final question for you to review Charlie's panel. And I want you to look at the fluorophores that Charlie has already designated and look at the stain index rankings and decide if any of them need re-evaluation. So the poll is open. Pop any answers in there. We'll give you a couple more minutes. Thirty more seconds to get any answers in. All right, so we've got some good answers here. All right, let's go through some of the answers. So we've got a lot of votes for PE size 7 being too bright, which you are correct. So we've got PE size 7 on CD19 for B cell exclusion, which is a highly expressed marker. So putting that on a super duper bright fluorophore is not only a waste because it's a dump marker, but it's also going to be way off scale. So we don't need to be putting that on such a bright marker. We also had a vote for BUV 496 being incorrect. BUV 496 is matched to our CD3, which we've got listed as a highly expressed marker, and BUV 496 is a really nicely dim marker. So that should work out well for Charlie's panel design. He's made a decent choice there. And we've also got a vote for BV 605 and 650 might be too dim for the antigens assigned. So those are both assigned to medium expressors. And yeah, maybe they could be slightly too dim, but they are sitting relatively in the middle, knowing that the top third from APC upwards here are actually really, really bright fluorophores. So somewhere in this range is probably pretty fine for medium. But yes, the one that was glaringly incorrect was that PE size 7 on CD19. Charlie should be using PE size 7 for one of his dim readouts rather than a B cell exclusion marker. Thanks for helping to design Charlie's panel. I'm sure he can now go forth with that to the cytometer and optimize it. But that's the end of the panel design section. So if you have any questions, please put them into the Zoom chat and we will answer them now. Or on Slido, we can answer them there as well before we go into our break. If there are no urgent questions, then feel free to keep typing them through the break and we can also answer them while we're on break. So we'll take 10 minutes off now and we'll come back at 2:15 to talk about
New Zealand time.
2:15 New Zealand time, yeah. 12:15 over in Sydney. Not sure what time it is over in Western Australia. Sorry, I don't remember all of them. But come back in 10 minutes and we'll start on panel optimization. But yeah, if you have any questions, feel free to chuck them in the chat and we can answer them at any point. All right, thanks guys. Welcome back, everybody. So we had some good chat in the Zoom chat about antigen classification, but we also had a question on Slido about where can you get a spread matrix for your flow cytometer. So for the Auroras, we're really lucky that Cytec has put together spread matrices for all of their configurations, which we can use. So those are available on the Cytec website or in the Fluorophore selection guidelines. For other flow cytometers, you have to generate one yourself, so you can run all of your fluorophores and then create the SSM in FlowJo. It's quite simple using their compensation wizard. So you'll have to create one yourself if you've got a cytometer other than the Aurora.
I think how to do it is explaining the protocol we published.
It might be.
Yeah. In the last protocol we published about panel optimization, I think there are the instructions of how to create the spread matrix in FlowJo by using your reference controls. Okay, shall we start? Cool. So let's start with the second section that is about panel optimization. And just to recapitulate after the break, or if some people are joining us now, we have designed already our panel and we have been following some important steps. And the steps are, first we need to have a clear experimental question. We need to know very well the biology or look for information. We need to know the instrument that we are going to use and the spectral signatures of the different fluorophores. And with the information that we have, plus the the cross-stain index matrix, the fluorophore brightness of all the fluorophores, at this point is when we can start assigning different fluorophores to each marker in our panel. And then the last step is to review if what we have put together can be run at the cytometer. We need to be happy. If not, it's better to go back and swap some markers. Sorry for this, we had some internet problems at the Malagan. So I was saying that I'll guide you through panel optimization, and that we have divided the protocol in 5 clear parts: antibody titration, a spectral reference control evaluation, and mixing evaluation of the fully stained sample, evaluation of marker resolution, and lastly, assessment of data quality. To start, let's talk about a very important point, that is antibody titration. Why is it important to titrate our antibodies? The titration of our antibodies is an essential step in panel optimization because it will help to improve marker resolution, so the separation between the positive and the negative population, also to reduce non-specific binding, to reduce the spread in high-dimensional panels, to use less antibody, and in consequence, to save money. When do we need to titrate our antibodies? It is recommended to titrate the antibodies when we order a new antibody, for example, or if we change from using one tissue to another one and we suspect that the antigen density can be very different. If the antibody lot changes, especially this is for tandem dyes, if we use a different instrument, for example, if we move from conventional flow to spectral flow, or if the experimental model we are using suffers a change that impacts also the marker abundances, or if we change the animal model. When titrating the antibodies, it is recommended to test different dilutions. The stain index is a very important calculation when titrating your antibodies, as it will help us decide the optimal titration to be used. The effective brightness of a region depends on the difference between the positive, the red histogram in this image, and the negative, black histogram populations, and the spread of the negative population. So, as Sam said before, if the median fluorescence intensity of the positive is lower, the stain index gets reduced. Or if there is an increase of the negative spread, as shown in this histogram, the staining index gets reduced too. It is recommended to use the stain index as a guide to select the best antibody concentration, and when possible, use saturation concentration. That is the point where all the targeted epitopes are bound to the antibody, and in this last image, where the green box is located in the image, this is a saturation point. Concentrations that are above saturation provides no extra resolution. There is instead an increase in non-specific binding that can produce artifacts in our data. We want to use saturation concentration, as this means that any change in fluorophore brightness that we visualize is a result in expression change of one given marker. Okay, here there's Charlie again, and Charlie needs your help again. Charlie, in this case, is titrating CD44 Alexafluor 700 antibody. And he knows that CD44 is expressed on all the granulocytes and myeloid cells in his sample. He puts the gate on only granulocytes and gets the resulting populations in his concatenated file, so with the different titers. We will start the quiz, and we will ask you what is missing and also how can we fix this. So we'll give you now 1 minute to join the quiz. And as soon as everybody is in the quiz, we will start asking you questions. So yeah, you have some seconds to enter the quiz to participate. You can use your name or a random name. We're waiting everybody to join the quiz. Getting there. Cool. Shall we start with the first question? What is missing in Charlie's titration plot? And you have 4 options to choose from. 10 more seconds, and we go to next question. Cool. Shall we go to next one? Okay, so we are showing the can you go back to the next question is, how can he fix the issue? Five more seconds. Cool. So let's go back to the first one. Can we go back? I don't know if we can. Okay, so the previous question was about what's missing in Charlie's titrations. And almost all of you got it right. So what he's missing is a negative population to be able to calculate the staining index. I think someone said that an unstained sample was missing. So if we have a look at the plot, the first sample on this titration plot is the unstained sample, where there's no positive staining. While when we have the different concentrations, there's no negative, negative population. Next question was about how can he fix this? And we got some suggestions. So some people say that it's a good option to put some cells, unstained cells, to each titration tube. So this way we will have a negative. And this one was not correct. So the correct one is include other cell types in the pre-gating. Why is not correct to include some cells in the unstained cells in each tube? This wouldn't be correct because we wouldn't be checking for a specific binding to the negative. Cool. And then, so the correct one is gating on more cell types. So if we gate only on the cell types, so we pre-gate on cell types that all express CD44, this is what are we going to visualize? Well, if the pre-gate, we make a bigger gate, including lymphocytes and other cell types, this is how the titration plot it's going to look like. And in this case, we will be able to obtain the MFI of the positive, the negative, the spread of the negative, and calculate the staining index to decide which is the best titration. So thank you for helping Charlie. And he has another question for you. This one is about he's titrating PDL1, which he knows is expressed on antigen-presenting cells. When he looks at his titration data, he has no idea where he should be gating positive and negative gates on the below plot. So in this plot, he should be able to gate on PDL1 positive cells. Our question for you now, in order to help Charlie, is what could Charlie do to make his titration gating easier? And I think, yeah, we're in a Slido. There's one open question. One more minute, we're getting some cool answers to help Charlie. Five more seconds. If you had a cool idea. Yep. I think we can talk about this. We got some cool suggestions. There's one option. Let's talk first about FMOs. The use of an FMO could help us see where PDL1 cells are sitting, but I'm not sure if in this case it would be enough to see where the positive, where they are, yes, but to get them out and to have a clear positive and negative. I don't have the FMO here to show, but yeah, I believe I would see where they sit, but it wouldn't be enough. There were other suggestions that this gating on the scatter profile, gate on APCs, and then from the APCs be able to see better separation, because we will only have the cells of interest that are in this case antigen presenting cells. This could work, but yeah, some other people say adding one more marker. And here I have the example. So in this case, what we are suggesting Charlie to do is to add CD11B, that is, it will be getting on antigen presenting cells. And from this, if we get on the CD11B positive cells, it gets easier to get on PDL1 positive cells. So this would be a good solution for him. Please, if there's something not clear enough, just pop up a question on the chat and we're happy to talk about it later. Okay, additionally, in some cases, we may require activation to boost numbers, and importantly, activation readouts should be titrated on activated cells. In this image, there is one example of CTLA4 PSI7 in stimulated and unstimulated cells. And as you can see, there are important differences when we compare the two titrations. Another important consideration that I was already starting to talk about in the previous slide is that we need to titrate the antibodies under the same conditions that the final assay will be performed. In this slide, the example is comparing two fixation and permeabilization kits for intracellular staining. And if we have a look at them, you can see that all the markers perform better under kit number 1 conditions compared to kit number 2. See, for example, Ki67, where 27% of the events were positive for kit number 1, while only 1.8% were positive for kit number 2 that we tested. Similar results were obtained for GATA3 marker and also for the other markers, intercellular markers in our panel. So therefore, for our panel, in this case, we decided to use kit number 1, and we need to titrate our antibodies under these kit conditions. All antibodies in the panel should be titrated, and this includes also viability dye. Some viability dyes, such as zombie NIR, are fixable dyes that react with primary amine groups on proteins. Live cells exclude these dyes, so that only cell surface proteins will be labeled, while dead cells allow the entry of these dyes into the cytoplasm, increasing the amount of total protein labeling. Dead cells will be significantly brighter for the zombie fluorescence than live cells. Viability dyes should be titrated to avoid positive staining of the live cells. And I'll show you here one example of one flow cytometry sample stained with zombie NIR viability dye. By gating the negative population, the intermediate, and the positive, we realize that all three populations have zombie NIR staining pattern. This is the result of not having properly titrated the viability dye. So we strongly encourage you to titrate viability dyes and validate that the live cells don't show a staining, such as in this example, where the negative population doesn't show positive staining for zombie NIR. And if you see a small fluorescence pattern in these live cells, it's because it's autofluorescence, not zombie NIR viability dye. Okay, so as soon as we are happy with our titrated antibodies, we can go into next panel optimization step that will be evaluation of the spectral reference controls. The first experiment that is recommended to be run should include single stained cells, so cells that are stained with each of the antibodies in the panel alone, but also single stained beads. And lastly, we need to run one sample with all the antibodies in combination on cells. Also, I'll give you one recommendation. After designing and optimizing several panels, I would strongly encourage you that instead of running your 30 or more color panel straight away, to start by optimizing a core panel. A core panel should include your main lineage markers, the ones that are essential to define the cell types that you are interested in. And then, as soon as the core panel is working fine, the remaining markers can be added. And if issues appear at this moment, you will know that they come from the additions and not from the core panel itself. So it's going to be easy to troubleshoot your panel. And also, a core panel could be shared between lab mates and only add some markers depending on your research question. Cool. So Charlie needs, again, our help. And at this point, Charlie decided to run his reference controls. And when he had a look at the data, he saw this in one of his tubes. So he gave it on the positive, gave it on the negative, and yeah, these are the spectral profiles that he could visualize on the aurora or in his spectral flow. I know he's running an aurora. What is going on here? Can Charlie use this control? So we'll give you now 1 minute to answer multiple choice questions. So 2 questions, 1 minute each. And yeah, let's start with the first one. What is happening in Charlie's reference control? "30 seconds left. Cool. Thank you for your answers. Let's have a look at your answers. So yeah, we have a big variety. So 60% of you say that his negative beads have high violet autofluorescence. 5% of you say that there is an issue with the cytometer. 20% of you, that is the correct answer, that his control is contaminated with another fluorophore. And 15% of you, he put the gate in the wrong place. So let's have a look at this next slide where I'll show you what's happening here. So in this case, when Charlie went to the cytometer, he ran first his BB421 control, and you can see how the BB421 control is looking like. But after he ran the BB650 control, and when the gates look like this, he's having a look at the positive population. But when he ran the gate into the negative, there are 2 spectra. One of the negative population and another one that is exactly the same to BB421 population. So this means that this control has been contaminated with, so BB650 has been contaminated with BB421. And yeah, Charlie will need to do something about it, because if he unmix his data using these controls, he's gonna have big issues, probably. So we have, okay, so now we would like you to type in the chat, what would you do if this happened to you? If you are running your controls and you are, the best practice is to have a look at the positive, but also the negative, because sometimes contamination hides in the negative. And what would you do if you are sitting in front of the cytometer and you see this issue happening? Yeah, please type your answers in the chat, either Zoom or Slido, because we are monitoring both of them. 1 minute. 10 more seconds to get some ideas for Charlie. You have to imagine him, you know, with a huge panel sitting in front of the cytometer and a bit stressed, because when you realize that this is happening, you really need to decide something fast. And cool, yeah, we got some answers. So there's one option. He can run into the lab and prepare a new control, but these are bits. These are bits, so bits can be prepared fast. But if he was using cells, going to the lab and preparing new cells would be a bit more tricky. There's another option that is using universal negative. So he can find a control where there is negative that is clean, and he can use this as a negative population. So this means that for the positive, for BB650, he would be using a nice clean BB650 control, and for the negative population, he would be using a clean negative. Were there any more suggestions? Yeah, I think this is everything. More ideas? No? Cool. Do we have another question? Okay. Okay, thank you for helping Charlie, and let's talk a little bit more about controls. As soon as we have acquired the data, at this point, we need to evaluate the controls, as we were saying. And the first important thing that I want you to bear in mind and keep in your mind for always if you're going to run full spectrum flow cytometry, and actually also for conventional, is that good controls probably is going to equal good data. So what are good controls, and how do we know if the recorded controls at the cytometer have high enough quality? Here, I'll give you some rules. First, positive and negative particles should be clearly separated. We need sufficient events for both the positive and the negative data points. We need to have been treated them the same way than the full stain sample. And they should have identical autofluorescence properties for the positive and the negative. Additionally, the spectral signature should be identical to the one in the sample. So if the signature of a certain control is different than the one in the fully stained sample, unmixing issues will appear. Here, I'll show you one example with CD19 BB711. In this example, bits and cells spectrums are overlaid, and there are small differences when comparing the two spectrums. When we have a look at the unmixing, when using bits is performing better than when using cells, using bits as a control. So, as the full stain are cells, we would need to use cells as spectral reference controls for CD19 BB711 to avoid unmixing occurrences due to spectrum mismatches from bits and cells. I hope it's clear. If not, yeah, just put some questions in the chat. And lastly, the last rule for the controls is that the spectral reference control should be at least as bright or brighter than the sample. In the first example here, HLA-DR BUV395 is brighter in cells than in bits, so cells should be used for unmixing. While in this second example, PLZF BCF594 is brighter in bits, so bits can be used for unmixing. Careful though, bits can be a great alternative to cells when there is not enough expression of a certain marker on the cell to be used as a single stain control. But the spectral pattern should be like the one in the full stain sample. Also, it is important to note that the unstained cell control remains critical for a spectral unmixing and must exactly match the cell type and sample preparation protocol used for multicolor samples. So if we are using a spleen, the unstained sample should be a spleen and following the same treatment than the full stained sample. We need to make sure that the fluorophore have the expected spectral signature. In this example provided by one user, E-fluorophore50 was recorded instead of FITC. So this resulted in unmixing issues. And when this user provided us with his control, we realized that his FITC control was another control. Also, when we go to unmixing, it will be important where we set the positive gates. To reduce the unmixing issues, we would place the gate to the brightest events of the cells. And in this example, if we have a look at the last row, we can see that if positive gate gets placed to dimmer events, unmixing issues appear, while if it gets placed to the brightest ones, there are no unmixing issues. Aurora, or Cytec Aurora, has the capability of reusing reference controls by recording reference controls to the library or by re-importing all controls into your experiment. When working with high-dimensional panels, so 30 or 40 color panels, this is a huge advantage as it will save you time, only having to record your controls once. Importantly, only high-quality controls are stored in the library or reused. So if we have an issue of one control, we won't reuse it or put this control into the library. We will make a new fresh one if possible. These controls can be used for future time points because they will be normalized based on the instrument QC of the day. And it is important to run new controls if we change the antibody lot, especially for tandem dyes, or if some changes are made to the instrument. So for example, post-service engineer visit. In this slide, I am showing you one example from Cytec where controls from the library have been used up to 77 days after the controls were recorded. Interestingly, the unmixing results look very comparable when looking at each of these 3 time points. Sorry, it's quite small. I hope you can see that the unmixing looks fine. Okay, one best practice. Dead cells and live cells will have different autofluorescent patterns. So to follow the rules for controls that we need them to have the same autofluorescence signatures, we recommend to kill the cells either with high temperature or ethanol and stain half of them, and then positive and negative will have the same autofluorescence properties. Importantly, this is not applicable for titrations, as in this case, you really need live cells. Cool. So Charlie needs again your help, and I hope you can help him after explaining you about the controls. So here we have a table, and I'll explain a little bit what this table means. So we have 4 different fluorophores, and then next column, it's going to tell us bits and cells. Also, so we have recorded bits and cells for each of the controls, and we need to decide if either bits or cells can be used, or both, or none of them. So next column is the brightness. So the brightness is the same or is 1. So in the case of P side 7, cells are brighter than bits. And also the third column is if the signature of the control matches with the full stain. So if there's a tick, it means that yes, it matches. And the last column is talking about the fluorophore being assigned to a rare marker. So not many events available, or cells available. Cells, yeah. So now we'll take a quiz, 1 minute for you to join and 1 minute per question. But yeah, actually we'll give you, I don't know, a couple of minutes to think about which controls should Charlie be using. And we'll start the quiz. Cool. So are we joining the quiz? Perfect. 10 more seconds and we will start with the first control. Okay, let's go. First question. For the first control, what Charlie should be using? Beats, cells, or either? Five more seconds. Okay. Cool. And the answer for the first question is the third one, either bits or cells, and I'll tell you why. Okay, I'm going to try to use the pointer and okay, perfect. I hope you can see the pointer. Maybe red is not the best color. The idea is that for VV737, either bits and cells have the same brightness. When we have a look at the signature, it's exactly the same. So the spectral signatures match. And this means that if we use either bits or cells, the full stain won't show unmixing issues. So yeah, correct answer. Let's go to next control, that is VV711. Yep. Cool. Time. So we got the 6% for bits, 88% for cells, and 6% for either. And the correct answer is cells. The idea is that the brightness for bits and cells is the same, but the spectral signature, only cells will match with the full stain. So this means that instead of using cells, we use bits, and mixing issues will appear in the full stain. So we can only use cells in this example. Let's go to next example. Five more seconds. Cool. So for this question, we got 6% bits, 76% cells, and 18% either. And the correct answer was only cells. So if we have a look at the brightness, cells are brighter than bits. So the full stain will be brighter than bits. And this translates into only be able of using cells, because if you use bits, as they will be dimmer, we will get unmixing results, unmixing issues, sorry. Cool, and last one. Five seconds. Good. So in this case, we got 41% beads, 59% cells, and no one, so that both of them can be used. So the correct answer in this case, is cells. Regarding brightness, they are both of them as bright when we have a look at beads and when we have a look at cells. When we have a look at the spectral signature, we realize that beads have a mismatch. So this means that if we use beads for unmixing, we will get unmixing issues. And we can only use cells. But there's something else. These cells, or these, sorry, not cells, the marker is a rare event. So this means that we're going to run lots of cells and we won't get probably enough positive events. So now we would like you to type to the chat which ideas do you have regarding how to fix this issue, or what would you do if this is your case. So in case it's not clear, I'll ask the question again. We have one control that we need to use cells because the spectral signature of bits is different. And when we run our sample at the cytometer, we don't have enough positive events. What would you do? Suggestions to the chats, either Zoom or Slido. Some more ideas? Okay, so we got some suggestions. I think one suggestion was to use cells and bits, and we have already said that we are testing bits and cells, and we cannot use bits, and we already know this because the signature, the spectral signature of bits is going to look different than the full stain one. So the only option is cells. What happened with the cells? We don't have enough positive events. So we have different options. One option that I'm not sure if someone suggested, use another marker conjugated to FITC. So as FITC is not a tandem dye, we could use another marker, for example, CD4, that we know the expression is going to be very high, conjugated to FITC, and we would get a positive staining, and positive and negative will have all the rules that I already talked about before. Another option is to use a different tissue. So imagine that our panel is in a skin tissue, and there are some markers that it's very hard to get them, but we know that these markers are not skin specific. They can also be found in a spleen or blood, and at the same time that we get one skin from mice, we can get the spleen and get heaps of cells out. So we could use a spleen sample. Any other suggestions? Shall we continue? Cool. Let us know if this is not clear or if you have questions. Happy to have a discussion at the end. Cool. So at this point, we're happy with the quality of the single stain controls, and the unmixing of the fully stained sample should also now be assessed. Cytec recommends users to start always by using Cytec assay settings, as they have been previously optimized by Cytec. This is a big difference from conventional flow, in which we need to set voltages. So in Aurora, we won't need to set voltages. The criteria used to establish the Cytec assay settings has been based on optimized resolution of cells that have been stained with anti-human CD4. The aim is to preserve the spectral characteristics of each dye and provide enough range to accommodate very bright signals. So it is recommended not to modify your gains unless you really need to. For example, when using a reporter protein that can be titrated down and it falls off a scale, it's one case that you should modify your gains. In this scenario that you need to modify them, you should try to preserve still the spectral signatures of the different fluorophores as much as you can. Identifying if significant changes have been made to the instrument settings can explain abnormal events in your data. So checking the related QC report, if available, will show you the ideal fluorescent detector gains and laser delays, so any changes can be identified. The drastic changes to the instrument settings can impact a spread in the data, where it would not be predicted during the theoretical panel design. So this is only for optimization part. Before checking the unmixing of the full stain, we need to make sure that the cells of interest are in the right place regarding the scatter profile, and that the forward scatter area scaling factor has been well optimized. And it looks like a nice diagonal line, making sure that we don't exclude big cells below. We need to make sure that all the cells are on a scale, and I'll show you now one video of how this is done at the cytometer that Sam recorded a while ago. Let's see. The screen is messing with me. Sorry. Maybe we need to go back to mouse and not laser pointer. Yeah. Oh, it was working.
Okay, the sound's not working, so I'll talk you through it in live time. So we're just running some cells at the moment, and I can see on a forward scatter versus side scatter plot that all my cells are way off scale. So the first thing I have to do is set my gain settings, because this is an Aurora. On a conventional, you'd be using voltages to bring my cells on scale. So I'm just adjusting my forward scatter gain at the moment, slowly getting my cells on scale. I bring them down. Okay, my forward scatter gain is correct, but my side scatter is still too high. I've got cells coming off the top. So if I bring down my side scatter gain as well, I want to come a little bit lower. Now we can see a few more events. You can see that most of my cells of interest are on scale. So I'm going to leave those gain settings where they are. And then over on the right here, I have a plot with forward scatter area on the x-axis and forward scatter height on the y-axis. And what I'm looking for is a nice 45-degree diagonal population from the bottom left corner to the top right corner. And as you can see, my cells are kind of falling off down to the right-hand side. So I need to adjust my forward scatter area scaling factor to get a nice one-to-one relationship between these two parameters. So under my lasers setting, I can find my forward scatter area scaling factor parameter. And to bring my cells up to the diagonal, I need to bring my scaling factor down. So usually, because QC is run using small polystyrene beads that are smaller than cells, the area scaling factor isn't usually set quite right based on QC. So we usually have to adjust this each time we run some cells through the machine. And eventually I get my setting correct. I get it on a nice diagonal, and then I double check my gains to make sure that I'm still on scale.
Cool. Thank you, Sam. Also, it's important to search that the fully stained sample has no abnormal events present that would make it harder when we want to evaluate the unmixing. Ensure that viability dye has been included so that you can eliminate dead cells from the analysis, and also check for antibody aggregates, which will appear as incorrectly unmixed hyperpositive events in some parameters, which can create supernegative events in other parameters. This must be gated out before going into the analysis. As soon as the full stain sample has been cleaned, it is important to verify that a positive signal can be found for all markers in the panel by plotting cytoscatter against each fluorophore in the panel. And in the case of rare markers, an FMO, that is a fluorescence -1 control, and I'll talk more about this control after, will assist in the detection of real positive events for a given marker. At this point, if no positive population can be identified for a given marker, it is important to verify that the appropriate stimulation or other experimental conditions have been used. Cool, and Charlie's back. Let's take a look at one parameter of Charlie's fully stained sample. And the question is, can you see anything in this parameter that might be, need improvement? So we are looking at BB711 CD19 here. The other markers are not as important. And we have a word cloud opening in yeah, and a couple of minutes to give Charlie ideas of what's going on here. So can you see anything in this parameter that might need improvement? Cool, 30 more seconds. Cool. So thank you for your ideas for Charlie. There's one that I think almost everybody is talking about, that is the BB711 versus Pacific Blue. And I'll talk about this option after. Now, someone said something about the zombie concentration. And sorry, maybe we didn't specify, right? But when we are evaluating the unmixing of the full stain, we have already gated out dead cells, so that's why zombie here looks, I don't know, there's no actually positive population because, yeah, we have gated it out in the cleaning steps before. So, yeah, let's focus on BB711 CD19. Some people are talking about compensation issues. So you are right, this could be manually compensated, but with a full spectrum flow cytometry, we strongly believe, and from our experience, we know it's possible that by using the right control, this issue gets corrected. So instead of fixing this manually, we would use cells for unmixing instead of beads. And I'll show you here how it looks. So the only thing we have changed from the left, so the red square to the right, the green square, is using a different reference control. So in the first case, we've used beads for unmixing, and in the right side, we have used cells for unmixing. This means that in this case, Charlie, the only thing he has to do is to use the right control. And beads have a different spectral signature than cells in this case of BB711 CD19 marker, and that's why cells should be used. Cool. Okay. So at this point, and we've seen it with Charlie, check the unmixing by visualizing an N by N matrix for each fluorophore against all the fluorophores in the panel. Here I'm showing one example of one panel unmixed with HLIDR BUV395 cells. And as it can be seen, the N by N matrix, the unmixing outcome is looking mostly good, with positive populations sitting nicely in line with the negative ones and not bending or curving up above or below the line of interpretation. However, in the case that you can find some unmixing errors, we recommend reviewing the controls and making sure that the unstained sample is that we are using is the correct one. let's talk about the FMOs. So in multicolor flow cytometry experiments, a spread is seen across detectors, and Sam has already talked about this. This spread is fluorophore dependent and can impact resolution. Fluorescence -1 controls became essential to set appropriate gates, particularly when the expression is dim or a smear. So I would only recommend to acquire them for some markers, not for all of them, probably for the readout ones to make sure that we are gating on the real positive events. In this table, I'll show you which fluorophores should be added in each control or sample to make sure that you understand what is an FMO. In the unstained sample, we don't add fluorophores to the sample. So there are no fluorophores at all. In the full stained sample, all the fluorophores are added to the sample. While in the FMO, so the column in the middle, all fluorophores will be added -1, and it's the one of interest. In this case, the one of interest is BB421. When we visualize the plots of the samples, establishing the cutoff between positive and negative populations using the unstained control is not as accurate as using the FMO. The FMO control allows us to account for the additive effect of a spreading from multiple fluorophores into the marker of interest, resulting in a more precise gating of positivity. So usually, as I was saying, we don't run all the FMOs when we are optimizing a panel, because if it's a 40-color panel, you can imagine how much antibody. We only decide which ones depending on how much do we know the markers and how difficult it's going to be to set the gates. Or sometimes we've used FMOs if we are troubleshooting one marker or some markers and it's very hard to find the reason why we're having issues, then an FMO can assist with it. But I said 3, 4, 5 FMOs, not 30 or 40 from our experience. Another important topic that we always get questions about is autofluorescence. Every sample will have different autofluorescence levels depending on the metabolic state, the type of cells, or the type of tissue that we are working on. This will translate into different autofluorescence brightness and spectral patterns in the samples aimed to be analyzed. Regarding autofluorescence signatures, 2 situations should be considered. The whole sample has the same or very similar autofluorescence pattern, and we will call it homogeneous autofluorescence, as the one I'm showing now in the screen of samples with PBMC samples. Or alternatively, there are different autofluorescence signatures in the sample, and we will call this heterogeneous autofluorescence. See, for example, in the skin, where at least 3 different spectral patterns can be identified. The autofluorescence can be the cause of poor resolution and unmixing issues also if we don't treat it appropriately. To decide which approach we are going to follow, we would need to determine in which category our unstained sample falls, to either homogeneous or heterogeneous. For each of these situations, we can follow different steps. In the case of homogeneous autofluorescence, we will extract the autofluorescence background. Here, one example of a gut sample where the autofluorescence extraction improves the resolutions of some markers, such as Ly6C, BB570, by lowering the background of the negative population while there is no change in the positive population. In the case of heterogeneous autofluorescence, the spectrums will need to be treated as additional fluorescence tags, so as if they were new fluorophores in the panel. I won't talk further about this today, but if you need detailed steps about this, have a look in our published protocol, where we attach one video with the specific steps of how to do this. Cool. We are almost there. Next step is to check the marker resolution of the markers in our panel. And we have Charlie here again with an issue. Charlie looks at his full stain sample and saw only a very dim staining for TCR gamma delta. The question here is this okay? Can he analyze this data? And yeah, 1 minute for a multi-choice question. And I think it's open now. Yeah. So do you think the TCR gamma delta staining is good enough for analysis? Questions coming in, answers coming in. 5 seconds. Is this staining for TCR gamma delta enough for analyzing this cell type? Yeah. Cool. And 100% of you answer you that it's not enough separation to analyze this cell type. To evaluate marker resolution, single stain cells will be compared with the multicolor stain cells. And it is very important to know that taking the time to compare each marker will be the foundation of panel optimization. If a reduction is seen, the impact of this can be further investigated to identify if it will impact the ability to identify the populations of interest. In this slide, I'll show you one example of a fluorophore in a single stain sample compared of two scenarios of the same fluorophore, but in the multicolor tube. In a scenario A, the addition of all the other fluorophores is increasing the spread of the negative population, and this can impact this marker resolution. In the case of a scenario B, the MFI of fluorophore X gets reduced in the multicolor tube compared to single stain control, and this can also impact the resolution of this given marker. In next slide, I'll talk a bit more about these two scenarios. In the case of scenario A, by comparing the full stain with the single stain, we will know how much our markers get affected with the spread after adding all the other fluorophores in the panel. This first marker, CD45RA-BUB395, doesn't get very affected by the addition of all the antibodies in the full stain. While this second marker, that is CD57-FITC, receives a bit more spread. And the last one, CD56-BUB737, receives even more spread. The more spread they receive, the more their resolution can be affected. At this point, one should consider how much the spread is affecting the ability to resolve populations, and if it is possible to go ahead with all the selected fluorophore, or if there's need to change some of them in our panel. In the case of a scenario B, that was about the loss of fluorescence intensity of given markers in the full stain when compared to the single stain, any reduction in positive signal should be investigated. One explanation is offered by Jalbert and others' paper, where sequential staining of chemokine receptors were required to achieve positive signal in the full stain sample comparable to that of the single stain cell controls. Sequential staining, interestingly, can be used to improve the accessibility of antibody binding sites, and it can also be successfully applied to non-chemokine receptors, such as the one in the example, so TCR gamma delta, the one that Charlie is using. Here we can see what happened when Charlie applies sequential staining. And in this case, Charlie can well gate out TCR gamma delta cells. Now, yes, we are almost done with the panel optimization. I know it seems long, but as soon as you've done it several times, it gets faster. Last step is the assignment of the data quality by analysis techniques, such as manual gating and also high-dimensional data analysis algorithms. So the last step in our protocol is the assessment of the quality of this data, and first by using expert gating. So we want to make sure that our results make sense biologically speaking. So for example, that B cells don't express CD3. However, we need to be very careful if using expert gating, as some results will be hidden, as we won't be checking all the marker iterations possible. So alternatively, to determine whether co-expression of markers makes sense or to identify additional errors in the dataset, dimensionality reduction algorithms such as UMAP or TSNE can be used. The goal of dimensionality reduction algorithms is to take a set of points in a high dimensional space and find a faithful representation of these points in a lower dimensional space. So for example, in a 2D plane. How can we interpret them if you haven't seen them before? We can overlay, for example, the expurgating populations gates to see where the different cell types are located in the map, such as on this example, where, for example, pink is where B cells are. Or we can color the map by marker expression heat map, in which for each marker, blue means that the marker is not expressed, up to red, that means that the marker is highly expressed in a given part of the map. Dimensionality reduction algorithms can help us answer some questions when having a look at the data or QCing our data. So for example, they could be useful for checking how consistent are our replicate samples, or which differences can we observe between conditions, and regarding the cell type abundances, or for example, the marker expression profiles. So regarding our optimized panel step, we can determine whether co-expression of markers makes sense, or to identify errors, as I was saying before, by using these dimensionality reduction algorithms, and a scale marker expression can be evaluated. Markers that are usually co-expressed should be found in similar regions of the Disney plot. So for example, FOXP3, the one I'm showing now inside the red circle, should be expressed in a CD4 positive region, and not in a CD8 positive region. So at this point, you should be confident that your panel is ready to go. And wait 1 second, because Charlie still needs your help for this step. Charlie is assessing his data, and his gating, his expert gating, looks normal. So he's gating on B220 positive and CD3 positive. So he gates on the CD3 positive events, and then CD8 and CD4 events, and it looks fine. And then he decides to run one high dimensional analysis algorithm, in this case Disney, and he sees something weird. Our question for you now, or Charlie's question for you, is what's going on here? And you can type your questions, your answers into the Slido. Charlie is almost done with panel optimization and would really appreciate if you have some suggestions of what's going on here. Are you still here? 10 seconds and we'll give you the answer. We'll give Charlie the answer of what's going on here. Cool. So we got some answers, and we were asking, what is strange about Disney and why might it be happening? And some answers that we got say that CD4 is co-expressed with CD19. And yeah, this is what is looking weird in this Disney. So CD4 and B220 are, CD4 is T cell marker and B220 a B cell marker. So they shouldn't be co-expressed. And if you look at this big island in the Disney map, they are both red for both markers. So yes, something is going on. And then regarding why this is happening, we'll show you what has happened here. So this could be a compensation issue, but in this case, Charlie modified his COM matrix and he didn't check these iterations. So he didn't check by expurgating B220 and CD4 because he gated on CD3 positive cells. If we look at this plot on the top, we can see that B220 cells are spreading into BUB395. So this means that B cells will be positive for CD4 while they shouldn't be. As soon as we fix this compensation issue, that in this case, it was Charlie's mistake that he changed his compensation, the maps should look like the ones that are in this slide. So there's one island that expresses CD4 and another island that expresses B220. So B cells and T cells. So let us know if there are questions. And yeah, we have now time for questions, and we'll have a short break after the questions. Do we have any questions in the chat as well? Cool. We have one question. How do you determine if the bits do not have a signatures match with full stain? So the idea is that when we run the first experiment, we have bits, cells, and the full stain. And the cells will have the same signature than the full stain because they are both cells. So how we determine if they are different is by overlaying the spectral signature. So we can normalize the maximum peak to 1 of the spectral signature, overlay them. Usually we use Excel. And you can see how they are overlaying. I'm not sure if we have one. No. So you just need to overlay them by using Excel and normalizing the maximum peak of both to 1, and you will see if there are different. It's quite visual. If they match completely, you can use either bits or cells. If they are different, bits will be different, and then you need to use cells. You don't have another option. Any other question? Good. Cool. So we'll take a 5 minutes break. So we'll be back at 3:35 for New Zealand. And yeah, I don't know for the other. So 2 hours less in Sydney, and not sure about the other ones. Yeah. And yeah, see you in 5 minutes. We'll stay here. So if you want to ask us some questions, we're happy to help and answer them now.
Hello everybody, welcome back. So for our last segment of the day, we're just going to talk about a few extra considerations that need to be made if you're planning a longitudinal experiment. So a flow panel that you're going to be running across multiple batches of samples if you want to be able to analyze them together in combination at the end. So Charlie needs one more piece of help from us. He has been running some samples with his lovely optimized panel, and he ran some samples across different days and wanted to be able to analyze them together using t-SNE. So he ran a t-SNE across these replicate samples that he ran on different days, and this is the result that he got. And our question, or Charlie's question for you, is why does the dark green sample stick out so much? And we've got another word cloud for you to answer. Why would one sample stick out from the rest when samples are run on different days? Let's give Charlie some ideas of things he can work on. So I'll give you a couple of minutes to get some ideas in. Few more ideas for Charlie, help him out a bit. It's his last question. Thirty more seconds. All right, let's take a look at the answers that we've got. So we've got a few different ideas, and a few of them are going to come on up in the rest of my talk, but we've got a few ideas around differences in staining and differences in the instrument, which could be something that's happening. And I saw someone popped in there at the last second with batch effects, which is a general overall term for exactly what we're seeing with Charlie's data here. So let's kind of talk through what can go on with a longitudinal study and how we can avoid these batch effects. So when planning a study and the collection of data, understanding the analysis intentions is critical to ensuring the appropriate steps are taken to achieve these intentions. This is especially important with longitudinal experiments where data collected at multiple time points will be analysed together, as extra controls are required, and these must be present from the start, with no changes to the data collection protocol throughout the study. So let's say we have an analysis intention to compare PBMC immune profiles from different time points within a participant. So we're going to monitor their immune profile over 48 weeks and take PBMCs at different time points, and we want to be able to batch analyze these together. Because we're working with PBMCs, this is pretty simple. We can just cryopreserve everything and stain and flow it all in one batch, so we can avoid batch effects entirely. But let's say we have a second analysis intention. We want to compare between participants at the same time. And because we're running so many participants across so many different time points, it's simply not feasible to be able to stain and flow everything in one big batch, because we're only human, we're not robots. So then we have to be able to design our experiment with the knowledge that we're going to be running different participants in different batches, yet we still want to be able to analyze them together. So we have to design our experiment focusing on mitigating batch effects and standardizing collection for successful combined analysis. With this type of analysis, the big challenge is variation from batch to batch. When it comes to high dimensional data analysis, which Charlie wants to use on his data, algorithms can't tell the difference between a marker shift due to a real experimental effect and a marker shift due to a batch effect. Therefore, we have to mitigate batch effects before we get to the analysis stage. There are three main sources of variation that we have to take care of. The first being changes in sample prep. If cells are handled differently at different time points, it may cause changes in the staining profile. Antibody staining is a tricky source of variation, as even if the same panel is used throughout the study, small variations in the staining process, such as pipetting or tandem degradation, can give rise to batch effects. Lastly, variability in the instrument can impact day-to-day measurements and MFI output. So the easiest way to prevent variation in sample preparation, and really the only option you've got, is to first standardize your protocol for staining before you run any of your trial samples or longitudinal samples. So you've just been told a really great process of how to design and optimize a panel. So you want to make sure you've gone through this whole process on some tester samples before you step foot in the lab with your real experimental longitudinal samples. So you really have to have the process down pat before you start your study. And as you may have figured out by how many steps that we've discussed today, this process can take some time, but if you're doing a long-term longitudinal study, which can take years, then spending a couple of extra months at the start, making sure that your protocol is well standardized and well optimized, is a very small investment of time. So here we have an example of the importance of protocol optimization. So this was some work where the first set of samples were stained at 4 degrees, and the results are shown in the red histogram. They tried it again to do some optimization by staining one of the steps at 37 degrees, and the blue histogram is the result. And across most of the markers, they saw an improvement in staining when they did that step at 37 degrees. So taking the extra time to run an extra experiment before starting your longitudinal samples will allow you to set off with the best possible protocol that you can have. If you get halfway through your trial and decide that, actually, I want to do this step at 37 degrees, it's too late. You need to be able to minimize your batch effects. So you have to do the whole longitudinal study using the exact same protocol. So get this out of the way first. Knowing that antibody staining can be impacted by small variations such as pipetting or tandem degradation over time, a staining control is an invaluable tool. A staining control is the same sample stained and acquired each time experimental samples are run. Due to the sample being identical, any shifts in output can be attributed to alterations in staining. It is a good indicator if an antibody is accidentally missed out of the staining cocktail or used at an incorrect concentration, which may not be obvious in experimental samples, especially if you're working with disease states. So before starting the study, the staining control needs to be collected. So for this study, we're running PBMCs, so we can just collect a large batch of PBMCs and aliquot them out so that we've got enough vials for each batch that we're going to run, and then we can cryopreserve them. All samples need to be identical, so they should all be collected, aliquoted, and cryopreserved at one time if possible. And then when we're running samples, or when Charlie's running samples, he could take all time points for one participant and one sample from the control donor and stain and acquire these all together. The third area where variation can be introduced is instrument performance. Standardization of an instrument to achieve stable output over time is relatively simple, and most cytometry labs will be doing most of the job already. The idea behind this is to ensure that given the same input, the cytometer is giving us the same output. The primary way to ensure that this is being achieved is through running the daily QC or equivalent setup process inherent to the cytometer. These systems run hard dyed beads and adjust cytometer settings such that the same MFI output is achieved. So step 1 of instrument standardization is ensuring that QC is run each and every day that samples are run. This should, in theory, standardize the output that is achieved, especially when using a system like the Aurora, which has manufacturer-designed instrument settings that usually aren't adjusted by the user. As an extra layer of standardization, we suggest running an additional set of hard dyed beads within the experiment and check the positioning of one of the peaks to ensure it doesn't move over time. Any hard dyed bead can be used for this. If you notice any shifts in these peaks, the instrument settings should be adjusted to bring it back to the expected output. Once all of the previous stages are complete and you've got all of your batched data, you still need to figure out if you've got any batch effects. To get a fast answer of whether there are any batch effects in the data, just like Charlie did, you can run a t-SNE on the clean data from your control samples, run on the multiple batch days that you've run samples. So here we have Charlie's samples all overlaid onto the same t-SNE. And even if Charlie follows all of the steps that I've just laid out, we can still end up with batch effects. Because this is sometimes unavoidable, algorithms have been developed to remove them. Cytonorm is a published normalization algorithm, which we've used on Charlie's dataset, and it takes the staining control as a training sample to train a model of normalization, then applies the same model to the validation samples or experimental samples. If we process Charlie's data using Cytonorm and rerun the t-SNE, suddenly everything overlays a lot more nicely and the data is more congruent from batch to batch. So once this normalization algorithm has been used, Charlie can be confident that he can go forth and analyze his data using algorithm-based analysis because the batch effects are pretty much removed. And as another way of testing how the different markers perform, or if marker-specific issues appear, is to visualize the MFI before and after normalization for each of the markers across all time points or batches. What we hope to see, like what we have here, is that after normalization, the markers look much more stable over time once they have been normalized. And Charlie would just like to say thank you so much for helping him out with his panel design and optimization. His PhD has been very successful with your help, and he's ready to celebrate and have a party after finishing his PhD.
Cool. So thanks for your attention, and we would really appreciate if you can answer a short survey about the workshop. It will help us do better next time. So we'll put the link on the chat, and now we have some time for questions, so we'll put our camera on, and yeah, we're happy to stay around for a while if you want to discuss something or if you have questions about your particular experiment. So yeah, how can we put